Good morning, dear friends, dear colleagues. Great pleasure to be here. I was last year on sabbatical with Idan like 10 years ago when this beautiful building wasn't here. So I'll tell you a little bit about what makes us human from the point of view of the Allen Institute for Brain Science. So just as a brief introduction, we are an institute. We're quite unique. We're 330 people funded by one donor, Paul Allen, the co-founder of Microsoft. And we execute these large projects for 10 years. So it's a finite project for 10 years. In this case, we're focusing on two very large projects. One is cell type, to get a complete understanding and a complete census of all the types of cells there are in the brain in two species, and only in two species, mouse and human. The second large project, the second large team project is building brain observatories in behaving animals where in a very highly reproducible condition, we record from thousands or tens of thousands or now close to 100,000 neurons using optical technologies and using high-density silicon probe neural pixels recording. So as I've mentioned, we're studying two species that's germane to this conference, the mouse and the human, the normal lab mouse, C57 black 6J. So we're focusing on cell type. So we're trying to understand primarily in cortex, but we're now expanding it to the entire mouse brain and the entire human brain with NIH funding. We're trying to understand what makes up a cell type. So the idea is that of all the 86 billion cells in the human brain, there are types of cells, so we can simplify the problem, not by treating each of the 86 million as discrete units, but as classes of units. And we'd like to define them. You can define cell types using evolutionary criteria. You can define them using developmental criteria, which we've done. Here we're looking in the adult, either human or adult mouse, and we're trying to characterize them by three broad modalities. Morphology, including connectivity. We're spending a lot of effort on trying to get specific connectivity using electron microscopy, using patching, multi-patching, and using whole brain neuron reconstructions. By physiology in slice, and then by doing transcriptomics. Essentially, the problem of cell type classification is really quite similar to the problem of classifying species. You know, this is what biologists do since 3 centuries. They classify species. And so it's very provisional, it's pragmatic. Everything sort of is based on hypotheses to be tested rather than absolute rules, because of course in biology, unlike in physics, there are no real rules. So we typically, so I'm just going to use these terms. You can classify cell types at various levels of resolution. At the top level, you know, they're class, excitatory, inhibitory, non-neurons and subclasses, types and subtypes. And down here, we're talking about probably on the order of 1,000 subtypes of cells. So we use large-scale single-cell or nuclear transcriptomics. So this is a popular technique right now. You heard it yesterday already. It's a very popular technique since it allows very easy scaling. Depending on how deep you go, whether you use a technique such as drop-seq that can go which relatively inexpensive, can survey 100,000 of cells, or you go very deep. We use a deep sequencing technology, Smarter version 4, where we typically read between 2 and 8 million reads, and we get on the order of 10,000 genes per cell, so that's a much deeper one. It's also, unfortunately, much more expensive. Where we get a bunch of cells, and then we classify them into the various non-neuronal, GABAergic, and glutamatergic cell types. And so we've done this at a high resolution a couple of years ago in mouse V1. I'll be talking to you about a very deep study where we compare two regions. One is in the back, primary visual cortex. ALM is a secondary motor area, more towards the front. We're now doing together as a part of a large consortium involving many people in Europe and the US. We're developing methods around primary motor cortex to compare all the different sequencing methods using the same tissue source. And then we're extending it, as I mentioned, with 3 very large NIH grant. We're extending this to the entire mouse brain at a high resolution using a common coordinate framework. So we're doing this because A, it's a nice way, it's a relatively cheap, inexpensive way to high throughput classification of a lot of cells. We can study the conservation across different parts of the cortex and across species. And of course, we are in the business of building tools. So we can use, once we know cell types, once we've identified specific markers, we can build transgenic animals and make them available, as we do through Jackson, to the community as a whole. And so ultimately, we like to get, transgenic animals for each of these specific cell types. So we can, turn things on or off, put in GFPs, et cetera. So here what we did, this is 20,000 cells, 2.5 million reads, roughly 14,000 are from V1, 8,000 are from the secondary motor cortex. So here we have, a large number of types. The types itself, that really depends on the clustering, are there 55 or 57, and it's less important. These are the number of genes we pick up. So we pick up on average, a median 10,000 genes in excitatory cells. So what we found is the following. It's a beautiful, simple principle. So here what you can see is a fraction of these cell types we detect in the mouse. In blue indicates V1, and black indicates in ALM. So what this tells us, for example, of the so here, if you do the split, here we use an iterative hierarchical scheme. First, you get a whole set of non-neuronals. And what you can see, we find those in both motor cortex as well as in primary visual cortex. Then, if you the same story, if you look at all the glutamatergic types, we find roughly 60 different glutamatergic types organized in these classes: PV, somatostatin, VIP, and LAMP5. Each of which, for example, somatostatin, there are 14 different types of cells. Here we find those, with one exception, in both visual cortex and secondary motor cortex. While if you notice here, the excitatory cell types we find either in V1 or in ALM, but not in both simultaneously, again, with one exception. So in other words, the principle seems to be that the inhibitory cell types, on the order of 50 to 60, are common in different cortical areas, while the excitatory cell types, of course, partially they're defined by where they project to, they all have different transcriptional codes. And we can see this by doing, so we did several thousands where we did back injection, where we injected, let's say, in the thalamus, or in the basal ganglia, or in the pons, and then trace them up to visual cortex, and then extract the RNA from the cells. And so we can see these different cell classes correspond to different projection targets. So it's really very, very specific. So with these two exception, Cajal retios and 2,6b types, which we find in both areas, otherwise these are all specific to one area. Again, with one exception, this type is shared across the two cortical areas. Otherwise, sorry, this is only in V1. Otherwise, all the inhibitory cell types are found in both regions. Now, we've done the same in human material. So we work with two sorts of human material. One, we work with post-mortem. In fact, what we've discovered now over the last several years, which is sort of relatively obvious, we're moving to very short post-mortem intervals, where we work with area hospitals to try to get, let's say, victims of car accidents that have been consented by their next of kin, and then use that tissue where we get very high RNA quality, close, RIN numbers of 9. We also use fresh neurosurgical tissue. I'm not going to talk a lot about it since Hype, the person who pioneered that, will talk more about that later on. But so this is what we find if we do exactly the same smarter version for. So we have this pipeline that at one end either goes in mouse tissue or human tissue and does exactly the same processing. Here we're doing nuclear RNA rather than cellular RNA, since it's much more robust. We have trouble if we dissociate the cell, particularly from the post-mortem tissue. Often the cells won't be conserved, so we go to a much more robust nuclear RNA and do both compare intronic and extronic RNA reads. And there you can see roughly the same organization. The glutamatergic family, you find the same four families: LAMP5, VIP, somatostatin, PV, and then here these are the glutamatergic and the non-neuronal cell types. with the same principle that we can see if we go to different areas, in this case, middle temporal gyrus, because most of our neurosurgical tissue comes from there, and we like to compare the neurosurgical fresh live tissue with the postmortem tissue, and then primary visual cortex. So in fact, what you can do, you can now do very close alignment, computational alignment between the human taxonomy, transcriptional single-cell nuclear taxonomy, and the one that we derive from the mouse, which is on whole cells. But we've also done a detailed methodological study where we compare whole cell RNA-seq with nuclear RNA-seq. So we really understand the relationship between reading the RNA that either comes from the nucleus or from the whole cell. And you can either just classify, so you can put them all in one bin and then do some various, here we did PCA, you can see beautiful, the human and the mouse material, or you can just look at the dominant, the canonical genes for each species, and then you can see they cluster together. So here you can nicely see the co-clustering of, for example, inhibitory cell types between human and mouse. So they do line up. There are significant differences. So, for instance, we find that in some cases, marker genes are conserved across cell types between human and mouse. In other cases, they're not conserved. And so we find a large, so if we plot all the scatter of all the genes expressed in the specific cell types, anywhere between 10 and 20% of the genes differ more than a factor of 2 between human and mouse cells. So here, for example, once again, we do we try to do detailed alignment between mouse clusters and human, and certainly for a number of them, we can find exact one-to-one where we can define homologous types between the mouse and the human. As I said, we can identify these same types, although they may or may not have the same marker genes. So that's really very important, particularly if you're in the drug business, because if you do something, if you try to target a cell type based on your research in the mouse, it may or may not carry over into the human. But we have that sort of very detailed molecular knowledge now. We're also doing, as I mentioned, morphology. So once you remove the scale bar, looks very similar, doesn't it? So in fact, I did this. So as I mentioned, we have 330 people at the Institute. So we had an all-staff meeting where I showed 12 images of single neurons. These are biocytin reconstructed, either human or from surgical samples or mouse. And we removed the scale bar, and people are essentially a chance at telling which one is human and which one is mouse. That's not to say that you can't do a deep convolutional network that will tell a mouse a human apart, particularly if you keep the scale bar, right? Because the cells are roughly 2.5 times larger in the human and in the mouse. But if you remove the scale bar, stuff is very similar. We're doing now this, of course, we're only doing in mouse, where we're doing whole brain reconstruction. This is together with our collaborators at Wuhan University, where we use a technology called FMOS. We use sparse fluorescent material. So here are 6 cells. These are the dendrites. 1, 2, 3, 4, 5, 6. These are claustrum neurons. I'm just showing you an example here. So these are neurons in my favorite structure, the claustrum. And you can see these are the dendrites, and these are the axons that project, single axons project throughout the entire cortical mantle. We're also finding this now, we're doing this at large scale in layer 5 and layer 6 neurons in cortex, and you can see this massive, massive projection throughout the entire brain. So what we're doing here, so we have the animal, we do the sparse fluorescent GFP labeling. The animal is sliced in 14,000 slices and imaged every micrometer and then computationally reconstructed. And what you see there is a tremendous amount of variability, and it's not clear to what extent the taxonomy, the classification that you see in the transcriptomics is going to relate to this incredible diversity in the synaptic, in the axonal projection. That's a big challenge. And of course, right now, this is impossible to do in human, obviously. And then we're doing what sort of Henry and Felix Schürmann and others have done since many years now. We're doing in vitro single-cell characterization, where from both, again, the pipeline is sort of agnostic to that, where either we have human material that we get from the surgery, or we have We have mouse material that we inject. We have a standard protocol since many years now where we do electrophysiology using a standard protocol or a combination of standard protocols, and then followed by biocytin injection and then reconstruction. So this is a paper we've just, it's under review, it's on bioRxiv, where we find 17 different electrophysiological clusters. So as it's well known, we find fewer electrophysiological clusters. Henry Markham in their paper calls them E-types and M-types. So we follow this classification into T-types, E-types, or M-types. So we have 6 inhibitors. So these are the associated T-SNE plots. These are the different, there are 6 excitatory clusters in slice and 11 inhibitory clusters in slice. This is not in vivo. Then we do the classification of their dendritic tree and their axons. And then we end up with this classification in mouse V1, where we now have close to 2,000 cells, where we find 14 excitatory M-types, which includes layers, 25 inhibitory M-types. So there's a total of 35 morphological types. There are 16 electrophysiological types. And if you look at together the space of electrical types and morphological types, then in principle here there could be, here there's a possibility of 84 different ME types. We find 44, and of the 231 possible inhibitory types, we find 68. The density here corresponds to the number of different cells we find. So then we are recording following the lead of 2 people, Gabor Tamas, as well as Hype, who's going to talk next about it, who really pioneered this wonderful technique. So we are implementing that now with help of Gabor Tamas, and now in collaboration with Hype and with Idan here also. We've implemented this high-volume pipeline where once every week or sometimes every 2 weeks, we get pieces of brain, this big, or sometimes tinier, sometimes bigger, from typical medial temporal lobe, from both epileptic patients or from tumor patients, where the neurosurgeon has to remove a deep tissue tumor, let's say in amygdala or hippocampus, and then tunnel through the overlying middle temporal gyrus. And then we have this, we have all this specialized thing, this equipment. Within half an hour, this slide sits in our lab. And, half an hour before, this contains somebody's memory of their first kiss. And now we can record from the individual neurons. And we can do it for in this case, look, 74 hours. Unlike mouse tissue that we all know, after 6 or 8 hours, the gradients run down and the action potential diminish. You don't find synaptic potentials anymore. We can stably record for 3 days from them. These are the various properties, physiological property, input resistance, action potential. We can do this in excitatory neurons. We can do this in inhibitory neurons. And so we're now doing the same classification. I'm not going to talk about it since the next 2 speakers will. We can do exactly the same classification using the same algorithm that we're doing in the mouse. We can do in the human and then do the alignment. Now, what we're also doing is something that I first heard Henry talk about many, many years ago. In this paper, where they use sort of PCR, sort of, remember that, sort of 20th century technology. Based on this, on these 2 papers from the Karolinska and from Tolia's lab in Baylor, we're doing what's called patch-seq, where we do all 3 things in one and the same, right? Because what we have, and what most people have, on the one hand, with high-resolution transcriptional analysis. Then we do this M&E slice analysis. What you really want to do in the same cells, you want to go in, record enough electrophysiology in order to say which type of E cell it is. You want to inject it with biocytin in order to recover the morphology of the dendrite and the local axons, particularly of the inhibitory neurons. And you want to suck out the RNA. And here it really helps to go for the nuclear RNA. It turns out to be much more stable in order to do assignment. Where in the taxonomical transcription tree is this cell? So you can do all 3. This is now working at high resolution routinely. The yield is still low. It's around 30%, but we're constantly that's a nice thing with the pipeline. sort of we have a lot of people focus on optimizing this, and we're also doing this in the human. Now, just a few cautionary notes here. So we all like to believe that life is simple and that sort of this, what Plato called, carving nature as its joint is easy. And there's a simple alignment between transcriptional types, morphological types, E-type, and ultimately what we care about, functional type. Unfortunately, we have not found that to be always the case. So, as I mentioned before, there seem to be many fewer E-types and M-types as their transcriptional types. And we have to see how that plays out, particularly now as we discover in other people like the light sheet technology people at Genelia, that single axons can be 10 or 20 centimeters long. Imagine that. The mouse is less than a centimeter linear dimension, and we find axons that are 20 centimeters long, and they can have incredible diverse projections. So we don't know how that is going to work out. Here we have done in 3 species, because we are so perplexed by this finding. We did it in monkeys, in human tissue, and in mouse tissue, where we look at the best known, outside the retina, the best known functional cell type characterization, which is magnocellular versus parvocellular. So we know in the monkey that you can block either selectively the magnocellular or the parvocellular pathway, and the monkey will have very specific deficits, either in color perception or in the perception of motion. We know that there are 2 very different pathways. It's well known, 30 years of experiments, particularly in the cat, in the monkey. Well, so we're doing exactly what we do in cortex, we're doing elsewhere as well, and we're doing in the thalamus, and we find different glutamatergic cells. We find different glutamatergic cell in the condylocellular pathway. We find different GABAergic cells, but we cannot distinguish in none of 3 species using our current technology, which goes pretty deep. As I said, we get typically between 8 and 10,000 genes at 2.5 to 8 million reads. We cannot distinguish magno and parvo. We're not saying there isn't any transcriptional distinction. At least we can distinguish with great ease all these different, 50 different excited cell types in cortex. We can't distinguish them in the adult monkey, human, or mouse. So we don't know yet where that's going to go. Then we are also we have this sort of this setup again that we really were inspired by the setup that Rodrigo Perrin in Henry's lab did, the octopatching. So we have it all here now nicely in a robotic control. You can identify cells, click on it, and you have robots that move in to patch them automatically. In human and mouse tissue, because what we're trying to get is cell type specific, what we call W alpha beta. Right? We'd like to get the specific weights among all the different types of neurons. Now that's a big space, right? Because if there are roughly 100 cells, there could be up to 10,000 specific types of synaptic connection. We know they're probabilistic. So here we can do it in the mouse because we can build triple and quadruple labeled. So we can identify different cell classes and put different dyes, red or green dyes in it. And so we know precisely where we're recording from and can probe these at high resolution. In humans, of course, we don't have that yet, although we're now getting enough knowledge so we can do very specific viral injection with specific promoters in the human, but our resolution is lagging behind that, what we can do in the mouse. And so I wanted to end with something that I've argued for many, many years, that I believe human and mouse are much more similar than they're dissimilar. So of course I all like to believe we are unique, and the belief in human exceptionalism, particularly in Western society, has been profound by, in fact, most monotheistic religion have this profound belief in human exceptionalism. Of course, we all know as biologists, we're just another animal. And as I said, at the level of the hardware, if I give you a little tiny quinoa grain piece of cortex, and I remove the scale bar, as I mentioned before, it's very difficult to tell apart, unless you're really an expert neuroanatomist and stare at this every day, the different type of tissue. The main difference between us and mouse is a factor of 1,000. And maybe 3 times, remember 2 numbers, 1,000 and 3. So 3 is the cortical thickness, right? In human, it's roughly, between 2 and 3 millimeters, depends where you look. In mouse, it's between 0.6 and 0.8 millimeter, so it's roughly a factor 3 difference. And so our cortical sheet in the mouse, it's roughly a centimeter, a square centimeter. In us, it's 1,200 centimeter, 12, like a pizza. The main difference is 86 billion total, 71 million. If we just focus on cortex, which is the one that gives rise to consciousness, it's 16 billion versus 14 million. So it's really pretty much exactly a factor of 1,000. And here, sort of the way, this is one way to think about it. Mouse, human. This is, you won't know this. Well, some of you older will know this. This is a 6502. Who knows what a 6502 is? Okay. My boss programmed this. The first compiler, basic compiler that Bill Gates and Paul Allen wrote was in a 6502. This technology is one of the first devices made by MOS technology. It has an amazing 3,500 transistors on it. It has a clock of 3 megahertz. It's 8-bit path. This one here, the A10, which is now, of course, already old, 2 years old, A10 has 16 nanometer technology. This is like 4 micrometer technology, has 3 billion transistors compared to 3,500, and the clock is 2.6 gigahertz. This one talks, this one doesn't, right? But in terms of now you can say, well, where's the fundamental difference between these two? Well, they both use and not technology, NAND technology. They're both von Neumann machines. So at the architectural level, they're really very, very similar. Yes, this is 40 years later of relentless evolution and progression, but the basic principle is really very similar. And so what people people always ask me, well, what's the magic thing about human? What's this one special neuron, right? The one special neuron that only we have that somehow magically explains why we can reach to the stars and, and do beautiful music like we heard yesterday night. But the fact is there isn't, yes, there are all sorts of specialization. These are both highly evolved animals. The last common ancestor lived, the placenta lived around 65 million years ago. We're both highly specialized for our respective ecological niches, as Chris Walsh expressed yesterday very beautifully. We have cell types that mouse doesn't exactly have, but we, as I said, for most cell types, I think we'll find equivalent. The number of cell types, just like the number of genes. If you go back to the 80s, people said, well, now that we know this is 20,000 genes, oh, there's going to be 100,000 genes in human, because we are obviously vastly more complex, right? Well, ain't so, right? We roughly have the same number of genes. We know that. Same thing with cell types. We'll have roughly the same number of cell types here and here. The difference is here, the neurons are somewhat more complex. You have 1,000 times more, and you have more layers. Now, if you look at today modern computer architecture and deep convolutional network, you know that the number of layers really makes a huge difference to the level of abstraction, right? If you look at shallow networks that have 2 or 3 or 4 or 5 or 10 layers, or you look at modern networks like at Google where they have 100 or more layers, those networks are much more powerful, although the principle is really very, very similar. We're all nature children, and the hardware we have is very, very similar. And that's our team, and with that,
thank you very much. Yes, please.
So the obvious problem with this numerical comparison between mice and humans is that some mammals have more neurons than humans.
Yes.
Elephants being a classic example, but dolphins and whales being another. And as somebody pointed out yesterday, they're not here talking about us. We're still, so clearly it can't just be numbers that makes a difference in human intelligence.
No, so it's an excellent question. Yeah, the Haber porpoise, right? There are 36 billion cortical neurons. There are other things that we don't value much, at least as scientists, because we think it's all about the brain. So, for example, we have hands. They have flippers. They live in a very different three-dimensional environment that's much more stationary than our environment. The explosion of mammals that settled all possible niches that can fly, swim, and run only happened 65 million years ago after the dinosaurs was wiped out, and you had this radical change in environment. So it may be that the environment they evolve in is very static. They can't manipulate it, and so it condemns them to being 100,000 while we are 7.6 billion. It may not directly relate to this. Just like, by the way, again, was mentioned yesterday, Neanderthal actually had a brain that was 10% larger, right? The endocarp was 10% larger. So why is it if they had a larger brain and supposedly had more neurons, were smarter, we survived? Is it we were smarter? Were we more aggressive? Did we outbreed them? There are all sorts of reasons that may not have anything to do directly with our vaunted brain.
Marshall.
Christof, you applied this fabulous analysis to the cerebral cortex. Are we really sure that interesting things aren't also occurring subcortically? Perhaps perception of pain or maybe perception in general?
Okay, so yes, you're perfectly right. There are other neurons outside cortex, I've heard, and we're trying to do that. But if you do study, for example, amygdala, again, there's a lot of similarities in the neurons. Ultimately, I do not believe that in the amygdala or in the hypothalamus there'll be dramatic different types of neurons between the mouse and humans. From the clinical evidence, I can tell you perception, particular conscious perception, in particular the perception of pain, is a cortical property. You need your brainstem in order to enable consciousness, just like you need your heart to enable to pump blood through your brain, otherwise you'll be unconscious, we know this. Your heart has to beat, so there are all sorts of enabling factors, whether it's the heart or the lungs or the brainstem, that need to be present in order for the organ that generates conscious sensation, which is cortex, to function. That's what the current evidence seems to suggest. It's a consciousness, specifically it's a cortical property. That's not to say in other animals that are not cortical, like for example the bee, which have a very complicated mushroom body, you may not get conscious sensation in non-cortical structures, but at least in us it seems to be a cortical property.
Do we have any students' questions? No? I'll talk to you about it later. Okay, so thank you very much. Thanks.